So today, I'd like to talk about fatty acid synthesis. But before I do that, I'd like to talk more about how we can deliver the building blocks for fatty acid synthesis into the cytoplasm. So fatty acids, right, you can have either free fatty acids or you can have triacyl glycerides. These are the packaged form of free fatty acids, okay? So these free fatty acids can be delivered to the bloodstream, to the capillaries, and they can act as an energy source or a nutrient for muscles and for the heart. So there are three ways that we can actually get these free fatty acids into the bloodstream. So let's take a look at each one of those. So there are three ways that we can get free fatty acids into the bloodstream. When we eat food, the first way is from the diet. When we eat food, we actually obtain triacyl glycerides. These can be converted into chylomicrons, which can then enter the lymphatic system. And from there, they can enter the capillary beds. Right? They can enter the capillary beds. The second way that we can get these free fatty acids into the bloodstream is via adipose tissue. So these are the cells that actually store free fatty acids as triacyl glyceride, TAG. So these are adipose tissue, or adipose cells, okay? And when insulin levels are low and glucagon is high, they can actually release free fatty acids into the bloodstream. So release free fatty acid to blood when insulin levels are low. When insulin is high, they store free fatty acids as triacyl glyceride, and when insulin levels are low, they release them, and so you get free fatty acids into the bloodstream. So we get the exact same result as we had above. So again, we get these into the bloodstream. And the last way that we can get free fatty acids into the bloodstream is via the liver. So the liver is what actually makes it. So the liver is going to take glucose, and I drew it right here, and it's going to convert that glucose into fatty acids, and then into triacyl glyceride. And this triacyl glyceride can actually be packaged along with cholesterol, and these triacyl glycerides can be combined with cholesterol to form very low density lipoprotein. Okay? And again, these can then enter the bloodstream. So there's many ways that free fatty acids can enter the bloodstream. Okay? And once they enter into the bloodstream via the capillaries, right, they can be delivered into either the muscles or the heart, for example, which are very, very important tissues, as you guys already know. Okay? Now, I'd like to talk a little bit more about the actual process. So let me actually zoom a little bit on the liver let's bring this up here, and let me zoom a little bit on the liver. So this is a liver cell. And as you guys know, we have the cytoplasm and we have the mitochondria, which is made up of two membranes right over here. And so what we have in the cytoplasm is glycolysis taking place. And glucose, which is a 6-carbon molecule, is going to be converted via glycolysis into 2 molecules of pyruvate, which are 3 carbons each. Okay? And what happens is that this pyruvate in the cytoplasm can then enter the mitochondria via transporters and be converted into acetyl-CoA, right, via pyruvate dehydrogenase complex. So this pyruvate, which is 3 carbons, is converted into acetyl-CoA, which is 2 carbons. And this is really, really important because acetyl-CoA is needed to make fatty acids in the cytoplasm. But there's a problem. We cannot get that acetyl-CoA into the cytoplasm because there are no transporters for it within the mitochondria membranes. So we need to somehow get that acetyl-CoA into the cytoplasm, right? So as you guys know, this pyruvate, which enters the mitochondria, is converted into acetyl-CoA via this enzyme, pyruvate dehydrogenase complex, and this acetyl-CoA can undergo the Krebs cycle within the mitochondria to make energy, ATP, as well as FAD and NADH, which can then enter the inner mitochondrial membrane and undergo the electron transport chain to make more ATP. So let's take a look at this here. This acetyl-CoA is combined with oxaloacetate, which is a 4-carbon molecule, to form citrate, which is a 6-carbon molecule. And this citrate can then be converted back into oxaloacetate. So that's why it's a citric acid cycle, or the Krebs cycle, because we're making citrate, which is a 6-carbon molecule, when acetyl-CoA and oxaloacetate are combined with each other. So that's the first step of the Krebs cycle. But this citrate, when it's converted back into oxaloacetate, is converted into 2 carbon dioxide molecules, which were present initially in the acetyl-CoA. So this acetyl-CoA is actually these 2 carbons are left in the form of carbon dioxide. Now, what's really interesting, why I brought this up, is because this citrate can go from the mitochondria to the cytoplasm. We have transporters for the citrate within the mitochondria membrane. So when the citrate enters the cytoplasm, it can be converted back into oxaloacetate, which is a 4-carbon molecule, and acetyl-CoA, which is a 2-carbon molecule. And that's how we get the acetyl-CoA that we need for the fatty acid synthesis in the cytoplasm. This process is known as a citrate shuttle. So the citrate can go from the mitochondria into the cytoplasm and then broken back into acetyl-CoA and oxaloacetate. Now what's really interesting is that this oxaloacetate can actually be converted into pyruvate. So we go from a 4-carbon molecule into a 3-carbon molecule. And therefore we need to lose a carbon, and we lose that carbon in the form of carbon dioxide. And what's really interesting is that in this process we convert NAD+ into NADPH. And this NADPH is what's needed to make fatty acids. And if you recall, the pentose phosphate pathway that I talked about in the previous video was the other way that we can generate that NADPH. If you forgot about that pathway, you can check out my other video on the pentose phosphate pathway. But this NADPH is needed to make fatty acids. So again, what we have here is a very, very special process whereby acetyl-CoA can make fatty acids in the cytoplasm, which will be the topic of my next video, exactly how that happens. And for that, we need some ATP, and we also need some NADPH, this reducing power, which we get by converting this oxaloacetate into pyruvate. And then this pyruvate can, of course, go back into the mitochondria via transporters and make more acetyl-CoA and more citrate, which can then be converted back into acetyl-CoA to make more fatty acids. So again, this citrate is broken down into the building block, acetyl-CoA, the building block to make fatty acids, and into oxaloacetate, which will give us the reducing power, NADPH, when it's going to be converted into pyruvate. And then pyruvate is also needed because pyruvate is what's going to allow us to regenerate the acetyl-CoA in the mitochondria so that we can make more citrate in the cytoplasm and more acetyl-CoA in the cytoplasm to make more fatty acids. So I hope this makes sense. And remember, fatty acid synthesis is an anabolic pathway, so it requires energy in the form of ATP.